Okay, thank you so much. I really appreciate the invitation and the chance to speak with you today about the research that I've been doing for the past 12 years or so. Um, I wanted to start by just giving you a brief outline of what I'll tell you about today. I wanted to start with honeybee hygiene and tell you why I think it's really the most sustainable option moving forward for controlling pests and diseases in honeybees. Um, we tend to think of a colony as hygienic or not, but really, I think there's lots of ways to select for hygienic behavior. And the result of that is that not all hygiene is created equal. So I'll talk about that a little bit as well. I want to talk to you about some of my early work with chemical signaling of honeybee brood, and the development of UBO, or the unhealthy brood odor assay. I'll tell you a little bit about how we discovered these chemicals and the development of UBO, characteristics of honeybee colonies that score high on this test. And then we'll talk a little bit, since it is a genetics conference, I'm not a geneticist, but I wanted to tell you a little bit about a heritability study I did, and some of the variability that we see in the field and what we're doing to address that. I'll finish up by telling you, why the key points, I think this work is important, where we plan to go from here. We have lots of exciting future research planned, and then we should have plenty of time for questions. I know with the language interpretation and lots of detail that I'm going to go over, there may be questions. So I'm happy to stick around and answer any questions, clarify anything, if needed. So this is a license plate that I picked up at a honeybee conference several years ago. And, I got it because it gave me a little chuckle. Professional gambler, I keep bees. But, I didn't realize at the time how true it really was. It does a lot of times feel like a gamble to be a beekeeper. And the reason for that, there are many different reasons for that, honestly. So bees have lots of threats, lots of issues. We have problems associated with natural parasites and diseases. You are all, I'm sure, familiar with the usual suspects that are challenging our colonies. We have issues, I think, even more so here in the US, but everywhere in the world, there are issues of poor management practices. I think especially after COVID-19, people understand that this kind of beekeeping can be just a disaster for encouraging the spread of diseases between colonies. We have issues of poor nutrition. So here I have a picture of almonds for pollination. This is a major pollination event here in the US. But a lot of monocultures are pollinated by honeybees. And we also have natural loss of natural forage, which is an issue. So just natural habitat for native bees, but loss of nutrition because of loss of natural habitat for foraging of honeybees. And then, of course, honeybees are exposed to pesticides. And, this happens at the flower, especially in those agricultural environments, but it also happens when we as beekeepers put these miticides into our colonies for control of those mites. Despite the gamble, despite the difficulties, for me, it's all worthwhile being a beekeeper, being a bee researcher, not only because honeybees are important, but because I find them, and as I'm sure you do as well, absolutely fascinating. Honeybees, I like to tell people this picture is like me going into my beehive. I feel so absorbed when I go into my colonies. There's just so much to see, so much to look at, so many interesting things going on. And so I absolutely love being a beekeeper and being a honeybee researcher. One of the things that I find particularly interesting and fascinating about honeybees is their mechanisms of social immunity that they've developed. So they have these ways of essentially cooperating to improve the health of the colony. So one example of this is grooming behavior. So here you can see an adult bee grooming a mite off of her sister. They also have a behavior called brood comb fever. Essentially, this works in a similar way to fever in our bodies, where the honeybee hive can actually elevate the temperature of the brood comb to eliminate pathogens. Honeybees also produce propolis. This is made from the resin of trees. Um, and for a long time, beekeepers were selecting against propolis. It's a very sticky glue that's difficult to work with. You know, it makes my wood break. It can be somewhat cumbersome. However, recent studies have shown that propolis has many antimicrobial properties, and actually the volatiles coming off of propolis are very beneficial for eliminating and reducing disease in the colony. And then finally, honeybees have developed the social immune mechanism of hygienic behavior. And this is really going to be the focus of my talk today and is the focus of my work. So hygienic behavior is the ability to detect, uncap, and remove unhealthy brood from the colony. So if you look in the middle of the screen here, you'll see some individuals pulling out an unhealthy brood. And of course, this frame is out in the sunlight, so I can take a video. But typically this is happening in the dark, and the bees will pull out these unhealthy brood and remove them from the colony. And I really do think this is the most promising and sustainable option for controlling pests and diseases in the colony. We can compare hygienic behavior to some of the other ways we can control for mites and diseases in honeybee colonies. So, for example, some beekeepers use miticides, some beekeepers use organic acids, like formic acid. Some beekeepers use mechanical interventions. But we know that there are problems with these. So, for example, we know that miticides can contaminate hive products. And eventually, mites will evolve resistance to these miticides. So it's kind of like a continual arms race. The mites that are more resistant to the chemicals that you put in the colony will survive. Their offspring will be resistant. And so when we're using chemicals to fight mites, varroa mites, we have to constantly be coming up with the next compound to use, or alternating use of this in our apiaries. We also have organic acids. These can be temperature dependent, depending on which ones you're using. They can be tough on queens, and they can be tough on the brood. So you can only use them, for example, certain times of year, based on weather. There are also mechanical interventions. I put here a picture of a tired beekeeper, because I think, you know, beekeeping is a lot of work, and mechanical interventions can be very effective, but are quite labor intensive. Things like caging queens or drone removal. And so, you know, though all of these methods can work, they have their negative sides as well. When it comes to hygienic behavior, mites cannot evolve resistance to hygienic behavior. At least they haven't for thousands and thousands of years. We know that hygienic behavior is really not tough on the queens, and it's only tough on those brood that we want it to be tough on. It's only tough on those unhealthy brood that we want out of the colony. And then it doesn't have to be labor intensive. So maybe selecting for hygienic behavior in your colonies is more labor intensive. But once you have that trait in your bees, the really nice thing is that it's not the beekeeper, but the bees that are doing the work. And so that's my ultimate goal, is to have a bee colony that's able to really manage itself, manage its own mites and diseases, taking the load off of the beekeeper and putting that burden onto the honeybees themselves. Another thing I really like about hygienic behavior is at the colony level, hygiene is preventative rather than reactive. So a lot of times when we're using a miticide, we wait until our bees, the varroa level hits a certain threshold, and then we treat to bring the varroa level back down. The nice thing with hygiene is it's there from the beginning. In February, in March, when that brood nest starts to build up, the bees are already performing hygiene and removing those unhealthy brood. And so the benefit of that is you are able to prevent the spread of viruses and diseases in your colony rather than kind of reacting to a problem once it's already there. However, not all hygiene is created equal, and this is especially true when it comes to the varroa mite. So past studies have shown that really lots of different types of hygienic behavior are effective against things like chalk brood and American foul brood. You'll notice that both of these are deadly diseases to the honeybee brood. But varroa is, in Apis mellifera, is not killing the brood. And so we tend to see variability in the results of how effective hygienic behavior is when it comes to varroa resistance. And so I think the main reason for that is that there are many different methods for selecting for hygiene. And every time you use a different method to select for hygiene, you get a slightly different result. So a few examples, we have the freeze-kill brood assay, which I think is very similar to the pen-killed, which is more common in Europe. But the freeze-kill brood assay is relatively rapid. You do have liquid nitrogen that you have to deal with that can be difficult to get hold of. But I think both for freeze-kill brood and pen-kill brood, there's improvement in mite numbers, but you tend to get mixed results in the efficacy. So it doesn't necessarily mean if you have a highly freeze-kill brood hygienic or pen-kill brood hygienic hive that the hive can necessarily take care of its mite loads without any beekeeper intervention. Kind of on the other side of the spectrum, we have the varroa sensitive. This has a lot of different names depending on who you're talking to, VSH or SMR. We tend to see with this, using this Harbo method of suppression of mite reproduction, we can get really good mite control using this selection method. So you can get bees that are really able to limit their varroa populations and survive without intervention on behalf of the beekeeper. However, the selection method is quite labor intensive. So depending on the method used, between 200 and 1,400 cells per colony must be inspected. And so it's a very detailed process and labor intensive. So when I started this work, I saw that there was really a need for a fast, accurate, reliable method of selecting for varroa and disease resistance. So I started studying chemical signaling. We knew that hygienic bees could detect a problem in parasitized brood cells. So hygiene has been studied for several decades. we knew that they're better smellers, and they're better able to detect an issue inside a brood cell. But the question really remained, what are they detecting? So some people thought maybe it was the smell of the mite itself. Some people thought it was movement of the brood inside the cell, so it's agitated and moving. Some people thought maybe temperature was involved. But we wanted to look a little bit more at the chemical signaling, and we knew that hygienic behavior occurs not just in response to mite infestation, but to many different diseases. And so I was really interested in looking at these chemicals that are found not on the mite, but on the brood itself. And so the honeybee brood has this, essentially the surface of the brood, the outer skin, if you will, is called the epicuticle. And this outer layer is covered with something called cuticular hydrocarbons. These are a waxy-like set of chemicals that originally evolved for desiccation resistance. So insects in general are typically quite small. And so what that means is they have a large surface area relative to their volume. And so this makes them very vulnerable to desiccation, to drying out, because they have that large surface area. And so insects in general have developed these cuticular hydrocarbons that are kind of a waxy protective barrier to help keep moisture in. But with social insects like the honeybees, we know that some of these CHCs have been co-opted for use in communication. And so we wanted to find out whether any of these cuticular hydrocarbons were involved in communication related to hygienic behavior. So instead of going into details of what I did to look into this chemical signaling, I'm going to summarize what I found in my PhD work in a single slide. This is all published open access, so if you're interested in learning more about the details, you should be able to find those papers. If not, I'm happy to provide them. But I want to give a brief summary of what we found so that we can move on to the more applied aspects and how we can use that information as beekeepers. So what we found is that there are these certain compounds elevated in varroa parasitized and virus infected brood. So brood that stick, not just varroa parasitized, but also those that are infected without varroa parasitization. And I want to point out that I say the compounds are elevated. So when we started looking for these chemicals, we thought we may find novel compounds that were present on unhealthy brood, but not present on healthy brood. And that's not at all what we found. What we found was essentially all the chemicals were the same on healthy brood and unhealthy brood, except that a small subset of these hydrocarbons, when a brood was unhealthy, those hydrocarbons are elevated. So there's a higher quantity of those hydrocarbons in the unhealthy brood. This was really an exciting finding, and we also found, importantly, that the compounds associated with this unhealthy brood are different from the compounds that are associated with dead brood. And I think that's really important when we think about the different methods of selecting for hygienic behavior. Many of the selection methods use removal of dead brood. Here we're focusing on removal of unhealthy brood. And then finally, we found that these compounds could be detected by the nurse bees. So they can notice them in small quantities, they can detect them, and they are actually the triggers for hygienic behavior. So we did some comparisons with chemicals very similar in structure, but maybe one carbon longer, or the double bond moved to a different place. And we didn't see the same kind of reaction with those control compounds as we did with these active pheromones. So I essentially finished my doctorate with these findings, and we felt like instead of being at the end, we were at just the beginning. We were really excited about these findings. When I started this work, I really didn't have any thought of applied science. I was thinking more just fundamental science, how do bees communicate? But what we realized is, we have something here that may have applicability for beekeepers. And so we went on to get some more funding to continue the research. And what we wanted to know was, can these unhealthy brood odors, and you'll see I call them UBOs, and that's where the name of this test came from, UBO. Can these unhealthy brood odors be used to measure varroa and disease resistance? And so we made a mixture of these unhealthy brood odors. They're synthesized in the lab, so we don't have to kill brood to get them. Chemists can make them. And I like to point out this gray text here was about 4 years of my life, so it seems very simple. But it took a long time to figure out how to make these compounds, and also to find the right mixture of these compounds and figure out how to apply them to develop this test. And so what all that work essentially has culminated in is the development of this test or assay called the unhealthy brood odor assay. And so I want to show you briefly what that test looks like, how it works, and then we'll get into how to use it in the field. So what we have is this is a small spray gun that we use. Here is a glass vial with the pheromones, and it has the UBO pheromones inside. And so what we do is we use a small piece of plastic pipe cut away to isolate an area of capped brood. We apply the UBO spray to that capped brood. So there are typically about 45 or 50 cells in this test area. We then take the treated frame and return it to the colony. After 2 hours, we pull that frame back out and then quantify the amount of uncapped brood, uncapped or removed brood in that test area. So we're just looking for a percentage. And then, so essentially that's how the test works. The important thing is what happens next, and that's that beekeepers can then take the information they get from this test and make informed decisions about breeding and about colony management based on these results. So I want to show you what it looks like in real life. So on the left side, I'll show you a high UBO response, and on the right side, a low UBO response. At time 0, we spray the pheromones onto the capped brood. And then at time 2, we pull that frame back out and we look. We may see something like this on the left with a high UBO response, and something like this on the right with a low UBO response. So if you look very closely, you can see here there's still 1, 2 cells that are capped. So this colony scored a 96%. It wasn't a full 100, but very close. And then here on the right, we had only a 24% response. There are 7 cells that were uncapped that were previously capped. So what you do with these results really depends on your apiary, your goals. If it was me with the colony on the left, I'm certainly not going to treat this colony sight unseen. This colony is probably resistant and can survive the winter without any varroa treatments. I'm probably going to graft eggs, make splits, potentially sell this colony, this queen, as a breeder. On the right side here, this colony, unless it has a high grooming or some other varroa resistance mechanism, I'm going to assume that this colony needs monitoring. It's probably going to need treatment for varroa mites. Because I like to increase the genetics of high UBO colonies in my apiaries, I may remove drone comb from this colony to make sure that the drones that are in my apiary are coming more from the colonies with high UBO scores. And potentially I would re-queen this colony. So here I'll show a video of a UBO assay. This video was taken a month or two ago here in North Carolina, and just shows the bees working about 1 hour into the assay. So I scanned down here just to show that the other cells are capped, and the uncapping you see is just here in the test area. Okay, so I want to tell you a little bit about the characteristics of high UBO colonies. Why would you want to have a colony that scores high on UBO? First, I need to explain what it means to be high UBO colony. And so this is a graph from a paper we published back in 2021. We did a single UBO test in June on over 70 different colonies. And then we looked at the varroa infestation for those colonies in August. And so each of these points on the graph represents a different colony. And what we found when we looked at this comparison was that colonies that scored a 60% or greater on the UBO test really were able to keep their mite levels consistently low. And so here I show that same set of data, but we've just broken it down into colonies that scored high on the UBO assay and colonies that scored low on the UBO assay. And so what you can see is the colonies that scored 60% or greater had a mite infestation average of 1.6%. These were all completely untreated colonies. In the colonies that scored low on the UBO assay, we saw an average infestation rate of 7.2%. The really exciting thing for me with this finding was that not only do high UBO colonies have lower mite infestations, but the UBO assay is really able to distinguish the colonies that fall above and below that treatment threshold. So typically, at least here in the States, most beekeepers will treat their colonies when the mite load reaches 2% or 3%. And so what this data showed us was that the high UBO colonies are really typically capable of keeping their varroa mite levels below that treatment threshold. Now we took the same 72 colonies and we also tested them using the freeze-kill brood assay. And what we found is that the colonies that scored high on the freeze-kill brood assay also had a lower mite load than the colonies that scored low. But what you can see is the freeze-killed assay is not able to distinguish those colonies that are really able to control their mites effectively without any intervention as clearly as the UBO test does. We also looked at overwinter survival. So we found that in those same colonies, survival of the high UBO colonies was 41% greater than survival of the low UBO colonies. Again, these were all untreated. And we found that survival of the high freeze-kill brood colonies was also greater than the low freeze-kill brood colonies, but the difference just wasn't as big. So again, UBO was more effective at predicting which colonies could survive without treatment, could survive that varroa load. We've also done some work looking at viruses. So we find that high UBO colonies have significantly lower virus loads. So here I show colonies that have a high UBO score in orange. This is their virus levels. And if they got a low UBO score, they're shown in blue. So here you can see there's just really pretty substantial differences in the virus levels between high and low UBO colonies. And we found that, this was not true just for viruses that are transmitted by varroa, but there are also some viruses like cyanide virus, which are not typically transmitted by varroa, but that we saw a big difference in between high and low UBO colonies. And so this just goes back to the point I made earlier. UBO is based on unhealthy brood odors. So while it works to identify varroa-resistant colonies, it's not limited to issues associated with varroa. It's really able to honeybees that are responsive to UBO are able to better detect varroa and many other diseases, brood diseases as well. One of the really fun things in the last 3 years or so is that I've been able to get this assay out to other researchers and have other researchers start to do testing and see what they can find. And so one of the collaborators we've been working with is Dr. Algier from the University of Vermont. And her group has done some really interesting work with nosema. And they found that, having a high or low score on UBO really had no impact on the presence of some amount of nosema in the colony. So regardless of the score they got on the test, the same amount of colonies from high and low UBO categories had nosema. But when we look at how much nosema those colonies had, when we look at the load, the number of nosema spores per bee, what we found is that the low UBO colonies had significantly more, significantly higher nosema loads than the high UBO colonies. And this was actually really surprising because nosema is not a brood disease. And so they've done some really interesting work following up on this, trying to understand, why is it that high UBO colonies are better able to control nosema? And so I'm happy to share some of those findings if we have time at the end. But Sydney Miller is really leading this work and has done a fantastic job looking into this. As you probably know, varroa entered Australia for the first time, or really started to spread across Australia for the first time just a few years ago. And so I've actually been working with Corinne Jordan at Bee Lady Apiaries and Bee Genetics in Australia for the last 3 years or so. And this is a data set that Corinne sent to me looking at chalk brood and UBO response. And so again, here we see that high UBO colonies have significantly less chalk brood than those low UBO colonies. I won't go into the details of how we tested this. If you're interested, ask me at the end. But we do see that high UBO colonies have better long-term memory than low UBO colonies. So we did some tests with learning speed and memory. We found that bees were able to associate these unhealthy brood odors with a sucrose reward. They were able to learn the difference between unhealthy brood odors and control chemicals at the same speed. They were able to recognize those smells at the same rate 2 hours later. But when we came back and tested those bees' memory 24 hours later, the bees from high UBO colonies had a better memory of those unhealthy brood odors. So I think, this shows us that they're more sensitive to those smells, and they have a better memory. Now, I do want to talk to you a little bit about a small heritability study that we ran. We are hoping to follow up on this with some genetic work. But essentially, I wanted to show or to determine whether UBO-linked traits can be passed on from one generation to the next. So here in North Carolina, I established a high UBO apiary. So every colony was above a 60% on the UBO score. The average was 85%. And then 8 kilometers away, we had a low UBO apiary. The average score was 29%, and no colonies were above that 60% threshold. From each of several colonies at each location, I made 2 sister splits. So we had 2 daughter colonies that were sisters, and then I just took one of those nucleus colonies from each site and then switched them for mating. And so what we end up with is 4 different types of daughter colony. We had daughter colonies that came from a low hygienic mother and mated in the low hygienic apiary. We had daughters that came from a low hygienic mother but mated in the high hygienic apiary. This was open mating. We had daughter colonies that came from a high hygienic mother but mated in the low hygienic apiary. And then the ideal situation, we have the colonies that came from a high hygienic mother and they mated in the high hygienic apiary. So we took these colonies, we waited several weeks, and then we UBO tested those daughter colonies to see how they scored relative to their mother. And so the test that we performed at 7 weeks, this is what we found. So here we have the 4 different groups I just described. These 2 are the high mothers, so the mothers were high UBO scores. Here's the high mating yard and the low mating yard. And then we have the colonies that came from a low maternal UBO with the high mating yard and the low mating yard. We also ran tests at 6 weeks. You can see here the same trend, but lots of variability in the test. This was the repeat at 7 weeks. And this was a test done because these queens were not all the same age. We wanted to eliminate environmental differences, so all on the same day we tested every colony. Their ages were 10 to 14 weeks, and this is what we found. So the same trend regardless, but you can see really it's important to wait 7 weeks before testing. And so what we found is, with that low-high combination, the average daughter score was still 15% lower than the mother. Here in the best of both worlds, a high hygienic mother and high mating yard, the average daughter score was 9% lower than the mother. And so what this tells us is UBO is heritable, or UBO-linked traits are heritable, but they do seem to be recessive. So you have to work to maintain this trait in your colonies. There are daughter colonies that scored higher than their mother or as high as their mother. But there were also colonies that scored lower. And so it's really important to test those daughters. And these findings kind of reflect well a similar study that was done at the University of Vermont. Here they did some instrumental insemination of the colonies and looked at the daughter colonies' UBO scores. So here we have colonies that had both the mother and daughter were instrumentally inseminated. Here in blue, we see open mated daughter queens from an instrumentally inseminated mother. Here we have open mated daughters and mothers. And then in the last category, we have an open mated daughter queens that were open mated from low UBO scoring colonies. So essentially a negative control. And so you can see again, this is clearly a heritable trait, but not one that's easy to fix into your population. So the major conclusions from this study, it's important really to wait at least 7 weeks post-emergence to test your daughter queens. The reason for this is we believe that hygienic behavior is really something that bees do when they're about 2 to 3 weeks old. So to test a new queen, you have to wait not only for her to lay eggs, those eggs to emerge, but also for those worker bees to be the right age to perform the hygienic task. We found that the mother's UBO score is really important. This is really critical, but mating also matters. And regardless of who the mother is, what kind of mating or insemination situation you have, you really have to test those daughters to know whether or not they're hygienic. So I stress this to breeders across the US and elsewhere. If you're selling queens as hygienic, please test them and make sure they're hygienic. Otherwise, they should be referred to as daughters of a hygienic queen, because I think it gives hygiene a bad name when queens are sold as hygienic, but really they're untested and it's not known how hygienic they actually are. So just to give an overview, I've shown you that high UBO colonies have lower varroa infestations, higher survival, lower virus loads, no SEMA loads, and chalk brood loads, better long-term memory. I also have data on relationships with freeze-kill brood and Harbo scores, so these are positively correlated, and we know that it's a heritable trait. We have some preliminary data showing that UBO response is linked to cell recapping, and expression of specific odorant binding proteins in the antenna. This is something we're following up on right now that's really exciting. And we've even seen some evidence that mites in colonies, in high UBO colonies, have lower virus loads. So not just the brood in those colonies, but also the mites that are found there. I think there's a lot of other things we need to tackle. We have no data yet on European or American foul brood. I think tropolalaps is something many beekeepers are worried about, and we don't yet know if tropolalaps is susceptible to hygiene. That's something I think we need to explore. And of course, there's always new and emerging diseases. Now, I want to touch briefly on UBO in the environment. We do sometimes see some variability in scores from the same colony over time. And so we're doing a lot to try and understand why that is. It's not something that's unique to UBO. This is also true for freeze-kill brood, pen-kill brood, Harbo scores. If you test the same colony at different times, you can get different results. So the first thing we've looked at is colony size. We see no effect of colony size on UBO score. So we've measured everything from 1 or 2 frames up to a very large colony. And what we see, this is really important because not only do we not want to select for small colonies that have low honey production, but we also want to make sure that if we have an apiary with several nukes and several double deep large colonies, that if we test both of those colonies, we can compare the results, and we find that we can. We've also looked at bottom board type and the effects of bottom boards, solid bottom boards versus screen bottom boards. There seems to be no difference in the effects of those on UBO score. We've also looked at nectar flow. So we know with freeze-kill brood that nectar can have an impact on those results. We set up 2 groups of colonies that had roughly equal average UBO score. We tested them in June during a natural nectar flow. And then we took those same 2 groups of colonies and we tested them again in August during a dearth where there was no nectar flow or very little nectar flow. But what we did is we fed one group a 1-to-1 ratio of sugar to water for a couple days before the test. And then the other group remained unfed prior to the test. And so what we found is there does seem to be a seasonal effect, but feeding your colonies, if you really do need to test them during a dearth, feeding your colonies can help make those test results more accurate. But there does seem to be both a nectar flow effect and a seasonal effect. And then finally, we looked at smoker use. So a study from 1995 showed that smoke can reduce the responsiveness of the honeybee antenna for up to 20 minutes. That's no problem if you're going in just to check on your colonies, but if you're running a smell-based test that only lasts 2 hours, that could be a problem. So what we did was look at the effect of smoker use on the UBO scores, and we did find that, albeit relatively small, smoke can have an effect on the UBO test outcome. So we recommend, really using minimal smoke or avoiding use of smoke during testing. So to wrap up, I'll just tell you, why I think this work is really important. I think UBO has a ton of potential. It enables breeding. It enables verification of how hygienic queens are. This graph just shows average UBO scores for some different populations that I've tested. So here we have VSH stock that's been selected based on Harbo. This is an apiary where the beekeeper was selecting essentially from these VSH breeders and breeding daughters off of those. The typical US commercial stock really has very little hygienic behavior. Hopefully this is increasing over time as the trait becomes more well-known and more appreciated. And then these are results from the Australian varroa naive bees when we first started testing them about 3 years ago. And I'm happy to report that we are making progress in Australia. So the progress is slow, but I think the speed of the change is increasing over the generations. So I show here we have a slight increase in our average UBO score. But when we look at the percent of colonies that we're getting over 20% UBO score, that's really starting to improve. So I think we're going to see a faster and faster improvement in UBO scores over time. The other thing I really like about UBO is one of my goals is to really democratize the selection process. And I think UBO is great because it helps facilitate local selection. So I put a picture of some snowy colonies here because I was working with these Vermont beekeepers that get lots of snow. They really need winter tolerant bees. And one of their issues was they kept bringing up these varroa resistant queens from the south of the United States, and finding that even though they got that resistance they wanted, the problem was they were losing their winter tolerance. So it's really nice to be able to select for hygienic behavior within those locally important traits. UBO can inform management decisions. You can ID susceptible colonies, make decisions about when to treat, isolating your resistant and susceptible colonies from one another. I think we have the opportunity here to boost the sustainability of the beekeeping industry by reducing reliance on these treatments that are always susceptible to mites becoming resistant. And ultimately, the goal, of course, is to improve honeybee health and survival. And for me, as a biologist, as a lover of nature and pollinators on a broader scale, it goes a little bit beyond honeybee health. I think we also have the potential to decrease the spread of pathogens and parasites to other pollinators. This is something that we're hoping to look into starting next year. But we know that honeybees and native bees share resources. We also know that there's spillover of viruses from honeybees to these shared floral resources and to native populations. And there can be some back and forth as well, where native bees spread disease to honeybees. So I think as beekeepers, we really have a responsibility to keep our honeybees healthy, and that's because we want our bees to be healthy, but also because doing so may benefit native bee health. So there's lots more work to come. Right now, we're working on identifying some thresholds for specific diseases. So for example, I showed you this chalk brood graph earlier. And what we find is while the threshold for varroa resistance is about 60%, if you want chalk brood resistance in your colonies, it looks from this data like you may only need UBO scores of about 20% or higher. So for different diseases, I think there are different thresholds of UBO response that we need to be able to obtain in our colonies. So we're looking into kind of figuring out what those thresholds are for different diseases. As I mentioned, there's variability across the season. We're really trying to understand why that is on a biological level. What are the things that influence hygienic behavior and behavior responses in the colonies? So we've started tackling these questions, but we really have a lot more work to do in this area. We're working with odor and binding proteins. Hopefully, some work will be done through the USDA with tropolalaps. And we have big plans to work with native bee health going forward. And lately, my focus has been to get this technology into the hands of beekeepers. So it's great to have an innovation in the laboratory, but it really doesn't do any good if it just stays in the hands only of researchers. And so we got a federal grant to really move this into the commercial space, and have been working to really get this out there for beekeepers to use and have access to. We launched in January here in the US, and we have plans to expand now to Canada, Europe, Australia, and New Zealand, hopefully by 2025. So I'm working with potential distributors to get it there, and happy to keep you posted. So I just need to say a quick thank you. Tons of great beekeepers, brilliant minds Critical funders have all been involved in this work and supporting this work. And one of the things that I'm most proud of is, we have these wonderful support from the university, from federal agencies, the USDA, NSF, nonprofit agencies like Project APIS-M, but we also have even had support from local beekeepers who, really found our work valuable and who contributed, sales from finances from sale of honey to help us really push this research and this technology forward. So lots of people to thank, and I will stop there, and I'm happy to answer any questions.